So in this chapter, we're going to look at how the microorganisms that live in and on the human body can interact with our physiology. You may recall the Human Genome Project that they finished a few years ago. Now there's an offshoot from that called the Human Microbiome Project. And this is where they're looking at the genetic sequences of the organisms that colonize our body. And they've found more than 700 species so far, and some of which are in surprising places. We thought that there were places in the body that we call axenic. Your book refers to it as sterile. That's not really the proper term for living tissue. Axenic is a better word for that. But we've now have a new understanding of the microorganisms in the body. So even though there's a healthy relationship with a majority of the residents on our body, modern medicine, things like immunosuppression and chemotherapy, even our diet can affect which microorganisms are the more dominant forms in our body. And this is having a big effect on human health. So where do the organisms that live on the body come from? So again, we used to think that places like the womb was axenic. Now we understand there's actually microorganisms in there. Our previous understanding was that the baby was inoculated first through the process of passing through the birth canal, where that amniotic sac broke and that fluid would then wash the walls of the vagina and introduce those organisms into the nose and mouth of the baby as they pass through. So this is why now we understand that why there's a higher rate of things like asthma in babies that were born C-section, because they were not inoculated with the right kind of bacteria, which typically includes lactobacillus. That's the most dominant form of bacteria in a healthy vagina. And so those organisms not only colonize the GI so that the baby can digest milk, right, lactobacillus, but it also provides competition against bad microorganisms that might colonize that GI. And so how the baby is fed after birth also contributes, right? There's a lot of microorganisms in breast milk, as well as antibodies and the other components of the immune system that mother can pass to the baby. So now they're looking at things like adding probiotics to formula so that they can mimic that transmission of good bacteria into the baby. Otherwise, if a baby is in contact with a lot of people after birth, then those microorganisms that are on those people's hands and in their as they breathe and kiss on them, they're being introduced into that new baby. And so this was where we basically acquire the population of bacteria that we're going to have the rest of our life. So there's some terminology that goes with the colonization of organisms and whether or not they lead to disease. And so obviously you first have to start with contact. If you don't have contact with the microorganism, they can't contaminate the body, which is just being present on the body. Once they're present, they may or may not be able to colonize the body, which means they're actually going to start growing. Once they're growing, if they can establish themselves by bypassing some aspect of the immune system, if they're allowed to actually grow and establish themselves, we would call that an infection. Not all infections lead to disease. We really don't use the word disease until there's a loss of function in the body. And so the word morbidity is oftentimes used to describe that. So recall that not all microorganisms can cause disease. Those that can have to have a special trick to allow them to bypass the human immune system, and we would call those pathogens. And so how much pathogenicity they have is referred to as the virulence. So there are pathogens with low virulence, things like lactobacillus, right? We just mentioned those are good bacteria in the body, but under the right circumstances, they can be problematic. Those that are highly virulent would be things like the plague or rabbit fever. Rabies is considered a highly virulent organism. And basically what puts you at one end of this spectrum or the other is if you come in contact with one of these organisms at the top of the scale, you will most likely get sick. Those that are at the bottom of the scale, things like thrush, right? That is something that is very common in HIV/AIDS patients because they have a weakened immune system. But most people would not experience infection or disease when they come in contact with this yeast. In fact, it's a normal resident of the human body. And so this is what this virulence scale is about. It's sort of how likely a human that comes in contact with them would end up getting a disease. And so we've mentioned some of these as we learned about the anatomy of both bacteria and viruses and helminths in previous chapters. If they can stick, if there's a special feature about them that allows them to create biofilms, things like that, this adhesion is one of the most important virulence factors. So if you're something like a helminth, if you can just hang on to the lining of the digestive tract, that is your special structure, that is your virulence factor. Viruses and bacteria tend to have surface proteins that might be called a ligand. In bacteria, we tend to call these adhesins. Adhesins are usually referred to those things that allow them to make that biofilm, because of course bacteria is usually the organism involved in biofilms. And usually if they don't have these special tricks, you might come in contact with the organism, but it's probably not going to be pathogenic. And so, in fact, we would call those avirulent because they don't have the one special trick that would allow them to colonize the body and cause disease eventually. Now, sometimes the virulence factor is something that the organism produces. And so if it's a live organism that can be secreting things like enzymes, they might come in contact with the tissues of the body and maybe dissolve away protective structures like keratin in the skin or collagen in the skin. And that would allow those organisms to invade the tissues further and maintain that infection. If it's something that they secrete that interferes with the immune system, then that would be something that allows them to, again, maintain that infection. So some specific examples of these enzymes would be things like keratinase. This would be how ringworm manifests because it dissolves away the keratin in the skin. Mucinase might be involved in those organisms that cause some of the STIs, like BV or bacterial vaginosis. Those would be one of the enzymes that allows those bacteria to actually invade the lining of the reproductive tract. Collagenase enzymes, these allow those tissues to be easily invaded by things like Clostridium. So gangrene, for example, involves some of these enzymes. So those bacteria can continue invading the skin deeper and deeper. Some strains of Staphylococcus can produce a coagulase enzyme that basically allows them to hide out in a blood clot and sort of wait out the immune response until they then would secrete staphylokinase to then escape from that clot and then be able to spread through the body. Streptococcus species, some can also secrete something called streptokinase, which does the same thing. In fact, this one is so effective at breaking down clots that we've turned it into a medication called Streptase. That's an anticoagulant. They can give this to heart patients or AFib patients, those that are at higher risk for stroke and things because of a clotting disorder. In some cases, it's not an enzyme, but an actual toxin. And these are typically protein-based toxins that are secreted by living pathogens, again, that can directly damage tissues, some of which actually will go and trigger the immune system, specifically the cytotoxic T cells, which are the ones that actually come around and kill cells that are either virally or cancerous, virally infected or cancerous. And so it's really sort of self-inflicted damage in that case. And so some of these toxins are going to be referred to as super antigens because they cause our immune system to sort of overreact. And so when we go to talk about necrotizing fasciitis, or what they call flesh-eating bacteria, a lot of that damage is actually self-inflicted by our own immune system because they're overreacting to these substances that are secreted. And so the toxins may actually be produced at the infection site, but they are carried around the body in the bloodstream. And so we would call this condition toxemia. And so some of the toxins that we're going to talk about would generally fall in one of two categories. Exotoxins are going to be those that are actually secreted from live cells, and they would include things like hemolysins, right? Red blood cells are very rich in iron, which is an important cofactor for microorganisms. If they want to replicate, they need a lot of iron. And so if they can break down red blood cells with hemolysins, then they have access to that iron that they need to grow and replicate. If it's referred to as a cytotoxin, this is sort of a very generic word, but it basically will kill the host cells that it comes in contact with. Typically, we're talking about tissue cells. If it specifically interferes with action potentials of neurons, it would be considered a neurotoxin. If it interferes with the cells of the digestive tract, it's probably going to refer to as an enterotoxin. So things like cholera or E. coli, they secrete enterotoxins. Endotoxin, we've met before. This is actually that substance that's in the outer membrane of gram-negative cells. But endotoxin, if you recall, only gets released when those cells die. And that basically that outer membrane disintegrates. And so exotoxin and endotoxin are two different categories of toxins. Sometimes the virulence factors that they have are physical structures. So for example, the capsule, we talked about a glycocalyx. If it's in the capsule form, remember that it basically serves as sort of camouflage, and the immune system can't see it, that Trojan horse kind of effect. And sometimes it's that the sugar that the capsule is made of is something that phagocytes don't have affinity for, so it's not really physically slippery. It's just that the white blood cells don't have affinity for those sugars because it's something that belongs in the body, or it mimics something that belongs in the body. Sometimes it's a chemical that they use once they've been phagocytized. You may recall we talked about fusion inhibitors, things like tuberculosis. This is the virulence factor. It actually thrives inside of a phagocyte because it keeps the phagocyte from doing its job. Leukocidins, just what it sounds like. It's going to be a chemical that once it's consumed by a phagocyte, it actually secretes these things within and it kills the cell from inside out. And so staphylococci, streptococci, there are several strains that can produce leukocidins. So recall that not all microorganisms can cause disease. They actually have to go through a set of steps to get to that point. And so in fact, the rule of thumb is not all contact leads to contamination. And contamination rarely leads to infection, and not all infection leads to disease. And that's because we have an immune system, and it is very effective. And so we're going to walk through some of these things, like the portals of entry and how they attach. We just talked about some of those virulence factors, how they can develop or colonize the body and develop biofilms. That's required. If they're going to bypass the immune system, they have to have those virulence factors that allow them to do that. And so we'll kind of walk through these steps a little further. So how do the microorganisms get access? We call them the portals of entry. And there's 4 major portals that microorganisms can use. The skin, of course, since it's our superficial barrier, is one major portal of entry. But if it's intact, most organisms can't get past it. Some organisms, even if it's intact, can still bypass it because they have keratinase enzymes, or they can just burrow right through the skin. Mucous membranes are a little easier to bypass because they're living tissues and they're non-keratinized. And so, but even still, mucous membranes are protective, and some organisms can't get through them unless there's a breach. So for example, HIV. That virus cannot access mucous membranes unless there's a lesion present or a tear in the mucous membrane. So that's why if the presence of any STD that causes a lesion will increase your chances of transmitting HIV by about 10 times because that virus would otherwise be excluded from the body. It couldn't enter the body. But syphilis, on the other hand, is a spirochete, and that bacteria can literally just bore right through even an intact mucous membrane. And then we have 2 other portals of entry. We'll talk about these other 2 here next. So any microorganism that can cross over the placenta would be referred to as vertical transmission because it's going from mother to baby. And we mean even across an intact, healthy placenta. So we use this acronym TORCH to help us remember those organisms that can cross. Now, fortunately, even though they can cross, they rarely do. It's only about a 2% chance that these organisms will cross a healthy, intact placenta. So toxoplasmosis, we group several of them into this category other. So HIV, Hep B, chlamydia all fall in the other category. Rubella, cytomegalovirus, and any of the herpes viruses can cross the placenta. So again, fortunately, it's rare that they do. But again, if you have a tear or anything in the placenta, then that's a whole different category of pretty much anything bloodborne can cross over if there's a breach in the placenta. So here's a little bit more information on some of those diseases that we know can cross over from mother to baby across the placenta. So some of them have an effect during development. And so you'll see here some things like spontaneous abortion or, derailing the nervous system development and things like that. Others will actually set them up for lifelong infections. If we refer to something as parenteral transmission. Now, this is not parent. I know the root word there suggests from mother to baby, but it's completely different. What we mean here is anything that bypasses the normal barriers of the body tissues. And so, for example, if you have a needle stick, you're bypassing the protection of the skin. This would be parenteral transmission. Surgery, wounds of any kind, insects that have piercing mouth parts can transmit pathogens that way. So these would all be referred to as parenteral transmission. So how do microorganisms get from one host to the next? They use what we call the portals of exit. Many of them are going to be similar areas where portals of entry, but these usually involve some sort of bodily secretion or fluid or something like that as well. So any of the secretions, so blood, urine, saliva, semen, any of those things would be potentially infectious if the organism is in this host. But the most common way, of course, is fecal-oral route. So there are millions of microorganisms in even the smallest amount of feces. And so if you have poor hand hygiene after using the restroom, or more importantly, in places where you don't have proper sewage treatment, it gets into the drinking water sources and things like that. And even shedding skin can contain infectious organisms like smallpox, right? That was how that organism spread, even from just the scabs that came off of the infected individuals. So some microorganisms can only be transmitted directly from host to host because they can't survive very long outside of that physiology. And so for those that can, we refer to the reservoir. So places where they can actually be maintained and be viable in between hosts would be referred to as reservoirs. And so there's three general types, right? Animal reservoirs. Now, generally, of course, humans are animals, but we are referring to non-human animals when we refer to animal reservoirs. For humans, we would refer to them as carriers, and there's going to be various versions of a human carrier. And then we have the non-living reservoirs, things like food, soil, and water. So for animal reservoirs, any of those diseases that we can contract from animals would be called a zoonotic disease or zoonoses. So this word sylvatic, that's a root word that means forest. And so this is sort of where they got the word sylvatic reservoirs. So in humans, because our physiology is so similar to animals, this is why we're susceptible to animal diseases, essentially. So there are more than 150 known zoonotic diseases, including things like rabies and the plague. But typically how we acquire these organisms from the animal reservoirs are either direct contact. So again, that would be an example of rabies. You have to actually be bitten by the animal. Or come in contact with animal waste. So things like E. coli infections that are coming from contaminated food from a downstream from a livestock area or something like that. Eating infected animals and undercooked pork, things like that. We talked about tapeworms. That's typically how we acquire tapeworms. But the most common way is through blood-sucking arthropods, things like ticks and mosquitoes, because we're much less likely to notice the infected mosquito than we are for something like a rabid dog, right? Where it's much easier to avoid that rabid dog than it is to avoid that infected mosquito. So some viral infections we can get from zoonotic reservoirs would be things like rabies, the viral hemorrhagic fevers, things like Ebola, and those we know those came from animal reservoirs. Hantavirus, now we have the coronavirus. They've tracked that to some birds and actually maybe bats even from one of those live animal markets in China. And so we'll talk about some of these diseases as we go, but basically these are the animal reservoirs, some of which have a wide variety of animal reservoirs that can carry the organism, like rabies. Pretty much any mammal can carry rabies, whereas only certain species can harbor the influenza virus varieties, right? There's a large family of influenza viruses. Now, some of the bacterial diseases that we can get from animals include things like the plague, of course, from infected rodents, even anthrax, right? Most people think of anthrax as something we get through potentially biological weapons, but historically, it's actually a zoonotic disease. We would get it from our livestock, but now that we have a vaccine and we keep the animal reservoirs vaccinated, we don't see that very often, but it is in fact an animal source disease. Other things like trichinosis or tapeworms, we talked about some of these, but we'll meet most of these diseases as we go through the semester. So human carriers, we have various versions of what we refer to human carriers, but if they're asymptomatic, this is one of the most problematic situations because they're still moving around in the community and they don't realize they're sick and they're spreading the disease, which is one reason why those diseases that have asymptomatic carrier stages are some of those that we intentionally make vaccines for, because they're spread so easily in communities without the person knowing they're infectious. Tuberculosis is one of those. So we don't have a vaccine in this country. Other countries where this is much more common, they do have a vaccine, the BCG vaccine. But one reason why we surveil healthcare workers, why we test healthcare workers continuously for tuberculosis, is because they could be asymptomatic in carriers, and patients that have immunosuppression, if they pick up that bacteria from an asymptomatic individual, can actually contract the disease more readily. Certain strains of hepatitis, you can have asymptomatic carriers, like Hep C, right? You may not have any symptoms early on in the infection. Typhoid fever, right? So think about Typhoid Mary. She was famous because she infected hundreds of people with typhoid because she was a cook and she happened to have the bacteria in her gallbladder and she was asymptomatic. So she was using the restroom, not having proper hand hygiene, and then cooking meals for a lot of people. And so it was quite readily spread in that circumstance. Diphtheria. This is one of those infections that not only in the incubation stage are you infectious, but even after, if someone has taken antibiotics and gotten treated for diphtheria, they are still contagious 30 days after the antibiotic treatment has ended. And so this is problematic. Syphilis, right? You can go for decades carrying this bacteria with zero symptoms. After the initial symptoms go away, you may not even realize you're still infected. And so this is one of those that's problematic. HIV, of course, also can have a latent stage of decades potentially, so can be spread without knowing. So this is from your book where they refer to some of those other carrier states, right? We just talked about the asymptomatic carriers, but you can be an incubating carrier. This would be at the front end of the infection where you don't realize you're infectious. Convalescent would be on the other end of the infection where your symptoms are all gone, but you still are infectious. Chronic carriers like typhoid Mary we just talked about, or passive carriers that don't even realize it's on their skin. So for example, again, hand washing is really important for limiting the transmission of infectious organisms. If it's a non-living reservoir, we're talking about soil, food, and water. And very oftentimes these all three include fecal contamination of some sort, whether it's human sourced or animal sourced. Very oftentimes agricultural areas are very close together. So if you have a crop that lives downstream from a livestock facility where the fecal waste is getting into the water that then they use to irrigate the crops, then those vegetables will then have E. coli on the surface. And so if you don't clean them properly, you've been exposed to this non-living reservoir. Very commonly E. coli that gets introduced from poor butchering processes. So there's some evidence that 1 pound of hamburger contains pieces and parts from 100 different animals, potentially when you buy it in the grocery store from industrial level meat packing. That really increases your risk that 1 out of those 100 animals that was butchered, that even the tiniest amount of fecal waste that got into that meat, and then it gets ground up and just mixed in. So we talked about that previously as well. So how it gets from host to host or reservoir to new host involves these 3 types of transmission. So contact transmission, we'll talk about some subcategories of each of these as well. Vehicle transmission or vector transmission. These are 3 very different ways that organisms can be passed from host to host. For contact transmission, it can be one of these 3 varieties. So direct contact, just what it sounds like. Most of the sexually transmitted diseases are spread that way. Animal bites, so things like rabies would be transmitted through direct contact. Indirect contact very oftentimes involves a fomite of some sort, so what we refer to as an inanimate object that participates in the transmission of disease, things like drinking glasses, right? So sometimes this involves being in close proximity, both in time and space. So for example, if you're passing something like an enterovirus that is going to be a digestive infection, it probably needs to be passed immediately, right? Those viruses aren't going to survive very long on that glass. But other things like the cold and flu viruses, we know can actually survive on metal surfaces, for example, up to 48 hours. So you just have to pass through a space where someone sneezed on their hand and then touched the doorknob that you follow up the next day, and you've gotten contaminated. So this is why washing your hands is so key, because these forms of contact, you may not realize you've been in contact because it's indirect. Droplet transmission does involve a space proximity. So for example, if you share an elevator with someone and they sneeze, you're in that 3 foot range, basically, where you can get something like the flu virus directly through respiratory droplets, or pertussis, right? Whooping cough or strep throat. Those things actually require close proximity where you inhale the respiratory droplets from another individual. Now, vehicle transmission involves something that can carry the pathogen, right? Think of a vehicle carrying you from point A to point B. Airborne. So it's not the air itself necessarily, but the dust particles that the organism can then adhere to are going to be carried. This one does usually involve a distance of some sort. So if it can be transmitted through dust particles, things like tuberculosis, then we worry about air handling systems in a hospital setting. So if you have a patient that's not in a properly filtered room, then they can be a source of airborne vehicle transmission of those pathogens. Water can also be a form of vehicle transmission. So if the organism can survive in the water body, and then someone downstream ingests it, then that can be a problem. We talked about cholera being transmitted through the Broad Street pump because it was a waterborne disease. Food, we just mentioned things like your vegetables, if they're contaminated with E. coli. Undercooked meat, for example, passing tapeworms, could then be a form of foodborne vehicle transmission. Vector transmission, on the other hand, requires a living vector. So usually it's an insect of some sort, and there's two varieties of vector transmission. If it's mechanical, it's more of a passive type of transmission, meaning it just is picked up on their bodies. And so, for example, a cockroach crawls across the floor of the bathroom, picks up some E. coli, and then carries it to your kitchen and leaves it behind on some food item that you left out. As opposed to biological vector transmission, involves biting insects. So ticks, mosquitoes, fleas, things like that would transmit things like yellow fever and Lyme disease, things like that. So I always like to remember biologicals bite. Then we have nosocomial infections, or healthcare associated infections. These are those that a patient acquires while they're in a healthcare facility. So those that are referred to as exogenous would be coming from outside the patient's body. And so perhaps maybe they inhaled some pathogen from a neighboring patient that was coughing out a virus, or a staff member that didn't use proper hand hygiene transmitted something from the skin of one patient to the next. Endogenous would refer to something that came from inside the patient. So their own microorganisms were affected by maybe antibiotics that they were getting while they were in the healthcare facility, and that caused sort of a shift in the balance of power. And so organisms that may or may not be harmful then can take over. C. diff is one of those examples. Very oftentimes people are walking around with at least a few of those bacteria in their gut, but when they're on high dose antibiotics or chronic antibiotic use, then those bacteria can take over. Unfortunately, we also have iatrogenic, and these are easily prevented by using proper aseptic techniques. So if you have a surgery where the equipment used was not properly sterilized, that would be an iatrogenic nosocomial infection. So here are some of the most common forms of nosocomial infections. You'll see one of the top two here is pneumonia, and that's probably associated with the use of respirators. And if they're not properly maintained, that's a really high risk of transmitting organisms directly into the respiratory system. Surgical site infections. Obviously, having an open wound will predispose you to potentially infectious organisms. So the CDC institutes a list of universal precautions that is supposed to help us limit the transmission of pathogens within the healthcare setting. And so things like a tuberculosis patient, they would be required to wear an N95 mask, which has a small enough aperture that bacteria can't pass through, whereas a normal face mask would not be a sufficient enough barrier for that particular pathogen. But no matter which disease you're talking about, hand washing is still the single most effective way to prevent the transmission of organisms. And so it's kind of unfortunate now with our healthcare setting where we have a number of patients being cared for by a very small number of staff. And so it's sometimes impractical to wash your hands between every single contact. And so in some healthcare settings, they have rules where you can use the hand sanitizer at the door of each room in lieu of washing your hands, but you can only do that with, I think, up to 2 or 3 contacts before you're required to wash your hands. And so still, it's if you recall from chapter 9, not everything can be killed by alcohol or whatever the active ingredient is in that hand sanitizer. So when you wash your hands, you're not selecting for any particular kind of organism. You're physically removing it from your hands so that you're no longer the vector, essentially. So infectious diseases generally go through these stages. The initial one, of course, is the incubation period. So right after exposure, you might have a period of time where there's no signs or symptoms. And every infectious organism has a different, what we call infectious dose or infective dose, which is basically the number of organisms you have to come in contact with in order for them to sort of progress and cause disease. Now, some diseases have prodromal periods where you just start to feel some vague symptoms. So, for example, the common cold has a prodromal period of a few days where you start to just sort of feel funky, as opposed to the flu virus. There's no prodromal period. You have a short incubation period, and then you immediately go into an illness stage. Once the symptoms hit, they hit hard. And so you can see here now, look at the load of the microorganisms in the body. Now, what causes the viral load, for example, if it's the flu, to turn around? Hopefully your immune system has finally caught up to it and is starting to remove those viruses. If it's a bacterial infection, maybe you went to the doctor and got antibiotics that were effective. And so that hopefully will put you into the decline stage. And so the signs and symptoms will decline, as well as the number of organisms in your body. Now, some infections will go into what they call the convalescent stage. You can see here that there's still organisms in the body. So you may or may not still be infectious, but the signs and symptoms are generally gone. Now, in some diseases, that red line would never go to zero, right? If you have a chronic infection where the organism is permanently in your body, your book refers to the final stage, sometimes what they call the fifth stage, is the continuation stage. That would be equivalent of a chronic infection. And so at what point in these stages is a person most infectious? Well, it really depends on the disease. So for example, Hep B. Once Hep B is in the body, it can become a chronic infection, and you're infectious at all stages, even before you have any symptoms. And so even the tiniest amount of blood is infectious for Hep B. And so that's why they tell you, if you are in contact with a Hep B patient, that you don't share razors or anything that might even have a trace of blood that could get transmitted, because it is infectious. Measles, on the other hand, is infectious at pretty much every stage until you get to the convalescent stage. And so, as opposed to something like shingles, right? A shingles infection can be infectious, but only when you have the open rash, the open wounds associated with the rash. Now, obviously, shingles is the latent version of a chickenpox infection. So if you came in contact with that person for the first time, you wouldn't get shingles, you would get chickenpox. But you can actually transmit that virus from those rashes. Now, how a disease manifests itself is usually described as signs and symptoms. Symptoms are those things that only the patient can feel and can describe to you. So these are what we call subjective. These would include things like the sore throat, fatigue, pain. These are things that only the patient can tell you that they're experiencing. Signs, on the other hand, are objective, meaning they can be observed, they can be measured. Things like sore throat, which was a symptom, can be detected by the sign that goes with it, which is inflammation. You might see redness and inflammation of the pharynx. You may not know that they are in pain unless they tell you, but you could also have signs that indicate pain, like they're crying or they're wincing, or they have facial expressions that are showing you that they're in pain, right? Fatigue is a symptom, but lethargy would be a sign, meaning you can see that they're dragging, right? They're just not getting out of bed. They look very tired. Fever is clearly a sign. We can measure that, right? We can feel it. We can use equipment to actually measure it. Cancer very oftentimes is diagnosed through signs like having increased white blood cell counts. Long before symptoms start, you can have signs that are oftentimes indicative of things like cancer. Syndrome is how we describe a disease by combining the signs and symptoms that belong to that particular disease. So, for example, you may have a multitude of diseases that have a particular symptom, but if it's not combined with certain signs, then it must be a different disease. And this is why we have to look at both, basically. If you have an asymptomatic disease, that means it's subclinical, meaning you would not know it even exists unless you see the signs, right? Because the symptoms aren't there. And so this is, again, why we vaccinate against certain diseases, because they can be infectious long before their symptoms start. And so we call those subclinical infections. So some infectious diseases can leave behind what we call sequelae, which is basically permanent damage to the body. So certain forms of meningitis can lead to deafness. Poliovirus can cause paralysis. Certain strains of streptococcus can lead to what we call scarlet fever or rheumatic fever. Lyme disease, of course, it got its name from the city in Connecticut, Lyme, Connecticut, where a pediatrician was seeing a really high rate of arthritis in his patients, which of course we think of arthritis as a disease of aging, and so that was an unusual thing. That's how it got discovered. Of course, historically, smallpox would leave behind permanent scarring, and that would be a form of sequelae. Remember how we mentioned Koch's postulates earlier in the semester? Well, here we'll talk about it in a little more detail. This is basically the set of steps that Koch developed to then sort of pinpoint which pathogen causes which infectious disease. And so the etiology of infectious disease is trying to find the specific pathogen that causes that particular disease. So how this works, it's basically a step-by-step process. You have to isolate the organism from every case of the disease that you look at, and you should never find that same organism in healthy individuals. So then you would take that organism that you've isolated, you would grow it in the lab, you would culture it, and then reintroduce it into a susceptible, healthy host. And this is key because not all diseases can, or not all pathogens can infect all species, right? There are some diseases that are exclusively human. So for example, gonorrhea. There's, no matter how hard you try, you could never give a rat gonorrhea. It's only humans are susceptible to that bacteria. Now, once that susceptible host has contracted the disease, then they would need to re-isolate the organism from that secondary host. And once you find that same organism that you found in the original host, then you can actually confirm that that pathogen is responsible for that particular disease. Unfortunately, Koch's postulates aren't always successful, and part of the reason why they're not always feasible is because some infectious organisms, we can't figure out what they need in a petri dish or culture conditions. If you can't culture it, then you clearly can't move forward through those steps that we just looked at. Some diseases have more than one type of cause. So for example, liver cancer. We talked about how hepatitis B and D viruses can be the underlying genetic alteration that leads to liver cancer when we talked about things like the oncogenetic viruses. But we also know that genetics on its own can predispose certain people to certain cancers. And so very oftentimes when we're talking about multiple factors here, it means different pathogens can cause similar looking diseases. So when we say meningitis, that just means inflammation of the meninges. But there are multiple forms of meningitis. There are more than 50 bacteria alone that can cause meningitis. And so, when you have diseases that look very similar, it's hard to pinpoint which pathogen is responsible. Now, we also now know, because of the Human Microbiome Project, that many diseases that we thought was caused by a single pathogen, now we know are what they call polymicrobial, meaning there's more than one pathogen involved. A lot of respiratory infections we now know is actually a combination of bacteria and viral infections working together, especially if you have chronic respiratory infections. And so this creates a problem when you're trying to do this sort of pinpointing one pathogen to one disease. So some diseases we've blamed other causes altogether. So for example, peptic ulcers we always thought was caused by stress or alcohol, aspirin use, things like that. Now we understand it's a bacterial infection, and this species just happens to damage the lining of the stomach. And so those other factors are in fact correlated. They just exacerbate the problem. So even though they found the organism, they ignored it as the potential cause. Sometimes the disease, or I should say the pathogen, gets blamed for a disease that it doesn't cause. So it just so happens that Haemophilus influenza happened to be very common in all the patients where they were testing the flu. And it turns out, of course, we now know it's a viral infection, so it can't possibly be this one. But he actually got named for a disease he doesn't even cause. Now, we do know that Haemophilus influenza can be a pathogen. It can cause meningitis, but it does not cause the flu. And so it's unethical to use humans to go through the steps of Koch's postulates. And so we have some diseases that we've yet to put through the steps and confirm that that is the organism that causes it. So we don't always have a healthy, susceptible host for diseases that are not human. So for example, syphilis, up until very recently, I think it was 2018, they were unable to culture this bacteria in the lab. So they've just only recently figured out how to grow them in the lab in the first place so they can go through Koch's postulates. Now that's one of the oldest human diseases. We've known for a long time that that organism is always correlated with the individuals that get diagnosed, but we've never technically taken it through Koch's postulates. So epidemiology, we talked about how John Snow sort of brought this idea to light, where you actually want to look at populations and how disease is transmitted amongst people. So epidemiologists generally use these four characteristics. We track incidence, which is basically how many new cases pop up within a given geographic area and over a given period of time. And that's because diseases all have a different time course. Some last longer than others. So when we talk about this given period, it depends on the disease sometimes. Prevalence would be sort of a cumulative number. So because certain diseases last a while, if you're looking at weekly data and it's a disease that lasts for 3 weeks, you clearly have to consider how many of the cases that you're counting are previously infected or are they new cases. So that's why we have both incidence and prevalence, because they represent two different things. And then of course we would look at how frequently we see the disease, how widespread or how widely distributed the disease is. These are the four basic characteristics that epidemiologists use when they're studying an infectious disease. So in epidemiology, we also use these terms to describe the distribution of disease. So if a disease is endemic to an area, it's something that we might see just at moderate intervals, sort of a stable, expected infection that'll show up on occasion. If it's sporadic, meaning there's a few cases that pop up very rarely in an area, that would be sporadic. Epidemics are when you have an infection that shows up in large numbers in an area. If that epidemic then spreads to new areas where you have a large number of cases in a number of different areas, then that would be a pandemic. There are generally different types of epidemiologists. Descriptive would be those who go out during an outbreak and take careful data and do what we call contact tracing to try to find index case. Now, generally, this may or may not be the absolute first case of the infection, but it's the first one that actually brought attention to the outbreak. In some cases, the index case is impossible to find if it's a highly virulent organism. So, for example, Ebola virus. Sometimes that can be fatal within 24 hours. And so trying to go back and find who was in contact with who, with witnesses who are already dead, it's impossible to trace it back entirely. Analytical epidemiologists would be those who take advantage of data after the fact. So these are the ones where, again, it's unethical to intentionally infect people, but once you've been through the outbreak, you can use that contact tracing and that information to try to get retrospective information and learn something from it so we know about disease transmission and how to control it. Experimental epidemiologists are those who are actually in the lab running Koch's postulates on lab animals and testing the hypotheses of trying to decide which pathogen belongs to which disease, essentially. So fortunately, we do have agencies that are working to continuously monitor and limit disease transmission. For example, water quality testing that the Arkansas Department of Environmental Quality does. They look for E. coli. and report on those. The FDA looks for food safety issues. Now, fortunately, in today's modern age, we have pasteurization, so that has limited the spread of many pathogens in dairy products, but we still do have people that consume raw milk. And so, fortunately, not a lot of issues come up with that because typically people that are drinking raw milk, they're small family farms, but there are things like listeria that can be transmitted in raw milk. And so pasteurization is going to help prevent that. In some cases, if we have a food-borne illness of some sort, just having on the news indicating that there's a food recall of some sort can help people make healthful choices. Alerting the public that it's time for the flu vaccine and things like that can limit an epidemic of those types of infections. And so, of course, some cases of disease are just what we call reportable, meaning once they get diagnosed, it's responsible. The health department is responsible for reporting it up the chain. So, for example, the local department would be our county health departments. They would report to the state, which then would report to the national level organizations and even potentially on to the international level, which is the World Health Organization. And so these reportable diseases, what we call the national reportable disease, they are published in what we call the MMWR, the Morbidity Mortality Weekly Reports. And so there's a long list of organisms that we consider reportable. So here's part of the list of reportable diseases here in the US. And so obviously by reporting these diseases, not only can it track outbreaks, but it can help, the statistics can help us with controlling future outbreaks. And so when we say they're reportable, keep in mind this is the healthcare provider's responsibility, not the patient, to report these to those agencies we just looked at. And so, for example, when we say reportable, some of them like gonorrhea and salmonellosis, those require a report in writing because they're not sort of an immediate issue, whereas something like measles or pertussis, those are actually required to be reported by phone because those can spread much more quickly. And so in some cases, if it's a sexually transmitted disease, for example, the county or the state would then try to follow up with any contacts, any sexual contacts with those infected individuals to make sure that they are disease-free and that they get treatment if they are infected. And here's the rest of that list. So by no means do I want you to memorize this long list, but just be familiar with some of those that are in fact reportable. So, all right, that's it.